America's future as a science leader may depend on students like the ones you're going to meet tonight, teenagers from Lambert High School in suburban Atlanta. They may have just found a better way to detect and treat Lyme disease, which affects nearly 500,000 Americans annually. Their primary tool? The revolutionary gene editing technique known as CRISPR. And these CRISPR kids did it to try to prove they are the best in the world, competing at a kind of science Olympics in Paris called iGEM, short for International Genetically Engineered Machine. But to win, they would have to go up against teams from China, the rising power in biotechnology. In Lambert High School's lab, Sean Lee and his classmates are teenage genetic engineers, manipulating the building blocks of life.
And what we're going to do is we're going to move each of these samples into these mixes. And these mixes have everything we need in order to amplify our DNA.
It's currently amplifying.
Yes.
All right.
And we'll have to do this 3 more times for our different samples.
So you would kind of swipe across.
We went to check out the iGEM team's project for the big Paris competition.
We first decided to map out where Lyme is most prevalent.
Their presentation seemed more like a pitch from a biotech startup than a public high school science class. Along with Sean, senior Avani Karthik is also a team captain.
And so it's a novel way of CRISPR that detects. And so we have to create a guide RNA. And when that guide RNA is recognized, the protein gets activated and it collaterally cleaves or cuts everything around it.
That just went right over my head. This is light years beyond my biology class, where the high point was dissecting frogs.
So you're just going to measure out 50.
This is called synthetic biology.
A popular example of synthetic biology is golden rice, where you're able to engineer this rice to have the specific vitamins that you want so that it's more nutrient dense.
And this would sound like cliche, but it's just endless possibilities. So you can just do whatever you want with it, as long as it's ethically correct.
To compete at iGEM, you need to use synthetic biology to solve real-world problems.
Lyme disease cases have doubled over the last decade.
These teens set their sights on finding a better way to detect and treat Lyme disease, something that has eluded adult scientists for decades. Transmitted by infected ticks, Lyme can cause arthritis, nerve damage, and heart problems if left untreated.
One of the biggest problems with Lyme is the lack of being able to diagnose it. So a lot of people will go years, we've met someone who went 15 years without a diagnosis.
Current tests make it difficult to detect Lyme in the first 2 weeks when it's easiest to treat. Lambert's big idea for better and earlier detection was to zero in on a protein generated by the infection. And this is the process where you work with CRISPR.
Yes.
Using the gene editing tool CRISPR and a simulated blood serum, they were able to target specific DNA strands where the protein hides, then snip away extraneous genetic material to expose the protein, enabling them to detect it with a simple kit-style test like a COVID or pregnancy strip. Did you guys ever think, this is way above my abilities, way above my area of knowledge, or you just dive right in?
Yeah, so actually we did reach out to a bunch of different professors and stakeholders who gave feedback on our project, and they did tell us in the beginning that this might not be so feasible because you're trying to tackle such a big thing.
They were also tackling how to treat Lyme. Standard therapy uses antibiotics, but Lambert planned to use CRISPR instead, targeting the bacteria that causes the disease. To make it work, they had to build software to model how best to use CRISPR. So as a teacher, is it easy to teach them because they're so smart?
They teach me. Are you kidding? They are so smart that I can't keep up.
Kate Scherer is their biotechnology teacher.
Because they are still teenagers, so they're thinking so far out of the box. This project in particular, I warn them this is very high risk, high reward. I can't imagine any of this working, but I'm happy to help you as much as I can.
Oh my God.
Lambert's team had advantages beyond audacity and brain power. Their lab is college level, funded by taxpayers and donors. This is one of Georgia's most affluent, high-achieving school districts. The student body is majority Asian American. The iGEM team is entirely Asian American, children of immigrants. Lambert also is a sports juggernaut. You can't miss the signs. But the white iGEM lab coat might be the most coveted uniform of all. I understand that parents move to this district so their kids can get into the iGEM program. And not just from Georgia, but from around the world.
Yes. And there are families that move specifically thinking that, well, my kid will be in biotech and then they'll have a chance to try out for the iGEM team.
About 100 students compete for roughly 10 spots on Lambert's iGEM team each year. Applicants submit a project proposal, take a test, and face an interview. Having a special skill like engineering or coding doesn't hurt, and you have to be willing to put in insanely long hours.
I need that to work.
With a month to go, they hit a string of successes. That stripe showed they could detect Lyme as early as 2 days after infection, far sooner than the 2 weeks with existing tests. Senior Claire Lee told us they also saw promising results in treating the disease. Doctors will be able to use this to identify Lyme disease and perhaps treat Lyme disease.
That's the goal.
What do you think of that?
We're doing something in our high school lab that could potentially have a huge impact for like millions of people. It's not like we're just saying like, oh, I'm just doing this little thing that like it might help my grade. This thing could help save lives.
What they found is just the first step. Much more testing will be needed. But they had more immediate concerns. They raced to finish before the international competition in Paris, pulling all-nighters to write results, code, and build a website explaining their project for the iGEM judges. It takes all that to be best in the world, the award Lambert won in 2022. Why do you do it?
I like to win, and so a lot of this is a competition, so I like to win.
You think you have a really good chance?
I think it depends on what the other teams bring.
Hopes were high as the Eiffel Tower when the students from Atlanta landed in Paris at the end of October. More than 400 teams, a third of them high schoolers, were competing in iGEM 2025. Elite synthetic biology with a dash of nerd-proud culture.
My first time coming here, I was so overwhelmed.
This was Avani Karthik's third year at iGEM, and she was keeping an eye on the other teams. Is there a project here that has blown your mind?
Almost every single one.
One was just a few feet away from their booth, Great Bay in Shenzhen, China. Great Bay's project developed a new enzyme for treating indoor mold. Other high school projects included designing crops to grow on Mars and eye drops to treat cataracts. As iGEM teams filled the floor this year at the Paris Convention Center, one thing was easy to see.
In the United States this year, we have 14 high school iGEM teams. Asia has 120.
Janet Standevan runs iGEM's International High School Division. Before this, she taught at Lambert, created its iGEM program, and helped it win the grand prize in 2022. Are you thinking to yourself, this needs to be in every school?
Yes. Oh, that's been our goal since day one, is let's start with Georgia as a little test bid and see what we can do here.
She left Lambert 3 years ago after securing federal funding to take synthetic biology to high schools all across Georgia. But the Trump administration cut the money, claiming it fell under DEI, diversity, equity, and inclusion. A judge temporarily restored the funding, but Standevan told us she's not sure it will extend beyond 2026. What did you think when your funding got cut?
I was devastated, absolutely devastated. I was angry. And again, I think anybody that's involved in this work at the high school level realizes this is necessary work.
In Paris, Lambert presented its work on stage.
Caused by the genus Borrelia, Lyme is transmitted through the bites of infected ticks.
And behind closed doors, the team dressed up to meet a panel of judges, answering questions about their lab work and its real-world implications. Is this what you imagined it would be 20 years ago?
Hard to imagine this.
Stanford professor Drew Endy was one of iGEM's founders. Back in the early 2000s, Endy was at MIT teaching genetic engineering, and his students had a competitive streak.
So we started iGEM because the people who wanted to work with us were the 18-year-olds.
iGEM thrived as biotech boomed. But Endy warned Congress America's lead in the field is slipping, while China has made synthetic biology a national priority. How concerned are you that there are not more American participants in iGEM?
I mean, it's profound concern. It's urgent that leadership of the next generation of biotechnology has a strong presence in America and is represented by young American leaders.
Endy was encouraged by the work done by the team from Georgia and wanted to know more.
Like, how do you deliver it?
Yeah, so we want to deliver it through lipid nanoparticles.
Endy echoed what other scientists told us, that Lambert's project could be a major scientific breakthrough if further testing pans out.
This year, they appear to have developed a better diagnostic for Lyme disease than anything I've seen before. It's not only applicable to Lyme disease, but anything you could find in your blood.
As groundbreaking as their lab work was, Avani Karthik knew Lambert was being judged on everything, from their website to their software. How do you think you're going to do?
I'm hoping we finish top 10, but even that, I don't know. We'll see.
That would be top 10 out of 140 teams. Fingers crossed, arms
so
crossed,
knock on wood.
Knock on wood, everything.
We're here to celebrate each and every one of you for all of your hard work, creativity, and enthusiasm.
On iGEM's final day, it was time to see what a year of work had earned Lambert High School.
The nominees are...
It was a nail-biter. They were nominated in 5 different categories and kept getting edged out. They didn't win the grand prize. That went to China's Great Bay.
So the winners are...
But there was joy. They got to storm the stage for Best Software Tool.
Congratulations, great job, great job.
We thought our project was amazing.
Hold on just a minute. Your project was amazing.
We're very proud of it, but it's what the judges think at the end of the day.
You're in the top 10
Yes,
in the world.
yes.
Lambert was the only American school to finish in the high school top 10, along with 1 team from South Korea, 1 from Taiwan, and 7 from China. It's challenging to tell a story about something that's invisible to the naked eye and tricky to explain. But it's one we undertook because rarely does a discovery come along that could revolutionize medicine. It's called CRISPR, and it stands for Clustered Regularly Interspaced Short Palindromic Repeats. CRISPR sounds more like a refrigerator compartment than a gene editing tool, but it's giving scientists power they could only imagine before, to easily edit DNA, allowing them to reprogram the genetic code of living things. That's opening up the possibility of curing genetic diseases. Some researchers are even using it to try to prevent disease entirely by correcting defective genes in human embryos. We wanted to see for ourselves, so we went to meet a scientist at the center of the CRISPR craze. This is CRISPR.
This has CRISPR in it.
So this is what's revolutionizing science and biomedicine.
This is what many people are using in research and trying to develop treatments.
That's wild.
Yeah.
That little vial is igniting a big revolution that is likely to change the way doctors treat disease in the future. One of the brains behind it is baby-faced Feng Sheng.
And how about the scaled up RPA?
At 36, he's already a tenured professor at MIT and a scientific celebrity because he figured out a way to override human genetic instructions using CRISPR. So the CRISPR is not the liquid. The CRISPR is in.
It's dissolved in the liquid. There are probably billions of molecules of CRISPR.
Billions.
In here.
In here.
That's right. And the way we use it is we take the liquid and we'll apply it to cells. It's very exciting, actually.
For the last 7 years, Sheng has been working on CRISPR at the Broad Institute in Cambridge, Massachusetts. It's a research mecca brimming with some of the brightest scientific minds from Harvard and MIT on a mission to fight disease. CRISPR is making medical research faster, cheaper, easier.
So just mix up these three.
Zheng's colleagues predict it will help them tackle diseases like cancer and Alzheimer's. How many diseases are we talking about that this could be used to treat?
There are about 6,000 or more diseases that are caused by faulty genes. The hope is that we'll be able to address most, if not all of them.
Most, if not all of them.
That's the long-term hope.
So we're talking diseases like Huntington's, sickle cell.
Yeah, ALS, hemophilia.
I think CRISPR, it's fair to say, is perhaps the most surprising discovery and maybe most consequential discovery in this century so far. How that code is read out.
To understand exactly what CRISPR is, we went to Eric Lander for a quick science lesson.
CRISPR, yeah.
He's director of the Broad and Sheng's mentor. He's best known for being a leader of the Human Genome Project that mapped out all of our DNA, which is like a recurring sequence of letters.
During the Human Genome Project, we could read out all the human DNA and then in the years afterwards find the misspellings that caused human diseases, but we had no way to think about how to fix them. And then, pretty much on schedule, this mind-blowing discovery that bacteria have a way to fix those misspellings appears.
This comes from bacteria.
This comes from bacteria. Bacteria, they have a problem. They came up with a real clever solution. When they get infected by viruses, they keep a little bit of DNA and they use it as a reminder. And they have this system called CRISPR that grabs those reminders and searches around and says, if I ever see that again, I'm going to cut it.
Zheng used that same bacterial system to edit DNA in human cells. Our DNA is made up of chemical bases, abbreviated by the letters A, T, C, and G. As you can see in this animation from Zheng's lab at MIT, a mutation that causes disease reads like a typo in those genetic instructions. If scientists can identify the typo, they can program CRISPR to find it and try to correct it. You program it. You say, I'm looking for this string of letters. And the CRISPR will go in, and out of all of the billions and billions and billions of letters on your DNA, find the exact ones that you have programmed.
That's right. CRISPR will allow you to do many different things. You can cut it, to edit it.
So you can snip out the bad part and you can add something that you want as well?
That's right. You can give the cell a new piece of DNA that carries the sequence you want to incorporate into the genome.
You say this so matter-of-factly. This is amazing.
It is pretty cool.
How many other labs around the world are working with CRISPR like this?
Many. One of the things that we have been doing is to make the tool available to researchers. To date, I think we have gotten it out over 45,000 times to 2,200 labs in 61 countries.
What are they doing with it?
They're using it to do everything. A lot of applications of CRISPR is really a Swiss Army knife.
Cue the worldwide CRISPR frenzy. At the University of California, scientists used a form of CRISPR to edit mosquitoes so they can't transmit malaria. Their colleagues are modifying rice to better withstand floods and drought. In China, scientists tweaked a gene in beagles to make them more muscular.
Those are reprogrammed.
A CRISPR vial from Shang's lab made its way to Dr. Kang Shang. He's an ophthalmologist and a professor at the University of California, San Diego, and wanted to see what all the hype was about. What did you think when you first heard of CRISPR?
I was a little bit skeptical.
Why skeptical?
It worked so well, too well, to be believable.
He decided to experiment on mice with retinitis pigmentosa, a genetic form of blindness. he conducted a vision test using a mouse with the disease.
This is the blind mouse.
This is a blind mouse. Then obviously you can see that he is ignoring the rotating stripes.
His researchers injected CRISPR into the eye of another blind mouse. The CRISPR was programmed to find the main gene associated with the disease and turn it off. It takes 3 months to see the results.
Now let's see how he's responding to the light.
He's following it around.
Yes.
Look at that. You're sure that he is seeing these lights.
This is actually a very commonly used test for vision.
How much of their sight do they recover?
About 30, sometimes even 50% of the sites for mice. They all have very strong research programs.
The next phase of Dr. Shang's research is to see how CRISPR works on one of our closer relatives. He sent us this video from his lab in China, where he's studying monkeys with retinitis pigmentosa. The blind monkey ignores the food. He says this monkey was treated with CRISPR, and it's easy to see the difference. Dr. Shang hopes to try this on humans soon. If CRISPR is used to treat disease or make a drug, it could mean big bucks. The Broad and Fengsheng hold a primary patent for CRISPR's use in human cells in the US. But no technology is developed in a vacuum. Biochemist Jennifer Doudna at the University of California, Berkeley, and her team made landmark CRISPR discoveries.
This is no longer science fiction.
This week, they are challenging Zheng and the Broad in court for the rights, arguing in part that Zheng's advance was derived from her team's breakthrough. It's a high-stakes battle. CRISPR is projected to be a multi-billion-dollar market in a decade.
Does that mean big business for you?
I think we're still quite a ways away from developing CRISPR into a real therapeutic.
I think you're being a little bit modest. I mean, this is sparking an incredible boom in biomedicine, and you're in the center of it.
I think there's still really a lot of work that still needs to be done. Developing the systems so that they are efficient enough, making sure that they are safe enough. But these are things that we're working hard to make possible.
But what if it were possible to stop disease from even occurring? That sounds like science fiction. But a team of researchers in Portland, Oregon, say with CRISPR, it's now a reality.
You correct it at the very, very earliest stages of life.
Right.
In the womb.
Even before that womb.
Manipulating embryos has been the focus of Shukrat Matalapov's career. He runs the Center for Embryonic Cell and Gene Therapy at Oregon Health and Science University. Matalapov is a maverick. He regularly makes headlines with his innovative, sometimes controversial methods to prevent genetic disease.
Preventing is always more effective, so there would be no recurrence of new disease, particularly when we're talking about heritable diseases that parents pass to children. And feeder has been taking off, right?
So Matalapov and an international team of scientists decided to use CRISPR on human embryos to correct a single genetic mutation that causes a deadly heart disease called hypertrophic cardiomyopathy.
We got lots of eggs, so that was great.
They got healthy eggs from donors.
Do you want the embryoscope dish?
And sperm from a man who carries the disease. At the same time the eggs are fertilized, they also get an injection of CRISPR.
Final co-injection?
Yes.
Matalapov enlarged the microscopic procedure over 300 times so we could see it.
Here we have our pipette with sperm inside, which has been already exposed to CRISPR. And this is an egg. And so what we need to do is pierce through, and then we break membrane, and now...
Release the sperm into the egg.
Yeah, and now this is the sperm coming in. Now it's inside there.
Just like that, that egg has been CRISPR'd.
CRISPR fertilized.
And you have changed the genetic destiny of that embryo.
Yes, we believe so.
These embryos will never be implanted, but they are grown in an incubator for 3 days and then checked to see if they carry the disease mutation.
The embryo work is...
Normally, 50% would. Matalapov says with CRISPR, 72% were free of the mutation that would cause the heart disease.
This is a huge advance in science and medicine.
We hope so. I think we're still kind of in the early stages. I wouldn't say that we're ready to go to clinics now.
He knows his results have to be replicated by an outside lab before they're accepted by the scientific community. But if they hold up, one day CRISPR could be used to help families that have been plagued by inherited disease for generations.
Is that what drives you?
Yes. Of course, it's a suffering of children, but also the guilt that parents have it, saying, I passed it to my child. So it's like, I caused this disease. And I think now we have a tools where we could help these families.
Metalipov wants to use CRISPR to eliminate disease, but the concern is his research has created a blueprint for less scrupulous doctors to design human beings, to edit embryos to make babies that are smarter, taller, stronger. Metalipov says that's not even possible right now.
Your critics say that you're playing God.
I think you could say to every treatment that the humans and doctors develop that we're playing God. God gave us brain so we could find a way to eliminate suffering of human beings. And if that's, playing God, I guess that's the way it is.
So what do you think about editing an embryo to prevent disease?
We don't really understand how complicated biology is. There's a gene called PCSK9. If you remove PCSK9, you can reduce cardiovascular disease, heart attack risk significantly. But it also has been shown recently to increase risk for diabetes. So how do you make the judgment call between these trade-offs? And there will likely be other impacts we haven't yet identified. So I think we need to wait and be more cautious.
I don't think we're close to ready to use it to go edit the human population. I think we've got to use it for medicine for a while. I think those are the urgent questions. That's what people want right now, is they want cures for disease.
Those urgent questions might soon be answered. A small clinical trial, the first in the U.S. using CRISPR to target certain types of cancer, is now enrolling patients.
I want to always balance hope versus hype here. While it's not going to affect somebody who might be dying of a disease today, this is going to have a real effect over the course of the next decade and couple of decades. And for the next generation, I think it'll be transformative.
Our lives have been transformed by the information age, but what's coming next is likely to be more profound. Call it the genetic information age. We have mapped the human genome, and in just the last few years, we have learned to read and write DNA like software. And you're about to see a few breakthroughs in waiting that would transform human health. For a preview of this revolution in evolution, we met George Church, a world-leading geneticist whose own DNA harbors many eccentricities and a few genes for genius. We found George Church in here.
Most of these are frozen George. Little bits of George that we've edited, all in different tubes.
Church threw himself into his work, literally. His DNA is in many of the experiments in his lab at Harvard Medical School. The fully assembled George Church is 6'5" and 65. He helped pioneer mapping the human genome and editing DNA. Today, his lab is working to make humans immune to all viruses, eliminate genetic diseases, and reverse the effects of time. One of the things your lab is working on is reversing aging.
That's right.
How is that possible?
Reversing aging is one of these things that is easy to dismiss, to say either we don't need it or it's impossible or both.
Oh, we need it.
Okay, we need it. That's good. We could agree on that. Well, aging reversal is something that's been proven about 8 different ways in animals where you can get, faster reaction times or, cognitive or repair of damaged tissues.
Proven 8 different ways? Why isn't this available?
What is available to mice?
In lucky mice, Church's lab added multiple genes that improved heart and kidney function and levels of blood sugar. Now, he's trying it in spaniels.
You did great.
So is this gene editing to achieve age reversal?
This is adding genes, so it's not really editing genes. The gene function is going down, and so we're boosting it back up by putting in extra copies of the genes.
What's the time horizon on age reversal in humans?
That's in clinical trials right now in dogs, and so that veterinary product might be a couple years away, and then that takes another 10 years to get through the human clinical trials.
Human trials of a personal kind made George Church an unlikely candidate to alter human evolution. Growing up in Florida, Church was dyslexic with attention deficit and frequently knocked out by narcolepsy.
What was it that made you imagine that you could be a scientist?
The thing that got me hooked was probably the New York World's Fair in 1964.
Welcome to a journey into the future.
I thought this is the way we should all be living.
Into the everywhere of tomorrow.
And when I went back to Florida, I said, I've been robbed, where is it all? And so I said, well, if they're not going to provide it, then I'm going to provide it for myself.
With work and repetition, he beat his disabilities and developed a genius for crystallography, a daunting technique that renders 3D images of molecules through X-rays and math. But in graduate school at Duke at the age of 20, his mania for the basic structures of life didn't leave time for the basic structure of life.
You were homeless for a time.
Yeah, briefly. It was
6 months?
6 months, yeah.
And where were you sleeping when you were homeless?
Yeah, I wasn't sleeping that much. I was mostly working. I'm narcoleptic, so I fall asleep sitting up anyway.
His devotion to crystallography was his undoing at Duke.
I was extremely excited about the research I was doing, and so I would put in 100+ hours a week on the research, and I pretty much didn't do anything else.
Not go to class?
I wouldn't go to class, yeah.
Duke kicked him out with this letter wishing him well in a field other than biology. But it turned out Harvard needed a crystallographer. George Church has been here nearly 40 years. He employs around 100 scientists, about half and half men and women.
Who do you hire?
I hire people that are self-selecting. They see our beacon from a distance away. There are a lot of people that are a little, might be considered a little odd, neuroatypicals, some of us are called.
Neuroatypical.
Right.
Unusual brains.
Right, yeah.
One thing about George that is very significant is that he sees what you can't even see in yourself.
Paris Tu Koshiklag and Alex are among the neuroatypicals. They're engineering human organ tissue.
I think he tries to promote no fear of failure. The only fear is not to try at all.
Corey Smith's project sped up DNA editing from altering 3 genes at a time to 13,000 at a time. Ariona Heisele went to Siberia with Church to extract DNA from the bones of woolly mammoths. She's editing the genes into elephant DNA to bring the mammoth back from extinction.
We are laying the foundations, perhaps, of de-extinction projects to come.
De-extinction?
Yes.
I'm not sure that's a word in the dictionary yet.
Well, if it isn't, it should be.
You know, there are people watching this interview who think that is playing God.
Well, it's playing engineer. I mean, humans have been playing engineer since the dawn of times.
The point is, some people believe that you're mucking about in things that shouldn't be disturbed.
I completely agree that we need to be very cautious, and the more powerful or the more rapidly moving the technology, the more cautious we need to be, the bigger the conversation, involving lots of different disciplines, religion, ethics, government, art, and so forth, and to see what is unintended consequences might be.
Church anticipates consequences with a full-time ethicist in the lab, and he spends a good deal of time thinking about genetic equity, believing that genetic technology must be available to all, not just those who can afford it.
This is the good one, yeah.
We saw one of those technologies in the hands of Alex and Paris-2 Koshiklag. They showed us what they call mini brains, tiny dots with millions of cells each. They've proven that cells from a patient can be grown into any organ tissue in a matter of days, so drugs can be tested on that patient's unique genome. You said that you got these cells from George's skin?
Yes. So we have.
How does that work?
Yeah, so we have a way to reprogram essentially skin cells back into a stem cell state. And we have technologies where now we can differentiate them into tissue, such as brain tissue.
So you went from George's skin cells, turned those into stem cells, and turned those into brain cells.
Exactly, exactly.
Simple as that. Organs grown from a patient's own cells would eliminate the problem of rejection. Their goal is to prove the concept by growing full-sized organs from Church's DNA.
It's considered more ethical for students to do experiments on their boss than vice versa. And it's good to do it on me rather than some stranger because I'm as up to speed as you can be on the risks and the benefits. I'm properly consented and I'm unlikely to change my mind.
We have a joke in the lab. I mean, at some point, soon probably, we're going to have more of his cells outside of his body than he has himself.
Church's DNA is also used in experiments designed to make humans immune to all viruses.
We have a strategy by which we can make any cell or any organism resistant to all viruses by changing the genetic code. So if you change that code enough, you now get something that's resistant to all viruses, including viruses you never characterized before.
Because the viruses don't recognize it anymore?
They expect a certain code provided by the host that they replicate in. The virus would have to change so many parts of its DNA or RNA so that it can't change them all at once. And so it's not only dead, but it can't mutate to a new place where it could survive in the new host.
Yes, he's talking about the cure for the common cold and the end of waiting for organ transplants. It's long been known that pig organs could function in humans. Pig heart valves are routinely transplanted already. But pig viruses have kept surgeons from transplanting whole organs. Church's lab altered pig DNA and knocked out 62 pig viruses. What organs might be transplanted from a pig to a human?
Heart, lung, kidney, liver, intestines, various parts of the eye, skin, all these things.
What's the time horizon on transplanting pig organs into human beings?
2 to 5 years to get into clinical trials, and then again, it could take 10 years to get through the clinical trials.
Thank you very much. Okay, wonderful.
Church is a role model for the next generation.
You got it working, it looks like.
He has co-founded more than 35 startups.
Oh, this is incredibly important.
Recently, investors put $100 million into the pig organ work.
Hey. How's it going? Hey.
Another Church startup is a dating app that compares DNA and screens out matches that would result in a child with an inherited disease.
You wouldn't find out who you're not compatible with, you'll just find out who you are compatible with.
You're suggesting that if everyone has their genome sequenced and the correct matches are made, that all of these diseases could be eliminated.
Right, it's 7,000 diseases, it's about 5% of the population, it's about $1 trillion a year worldwide.
Church sees one of his own genetic differences as an advantage. Narcolepsy lulls him several times a day, but he wakes, still in the conversation, often discovering inspiration in his twilight zone.
You could do a binding assay where you have like 2 fluorescently labeled cells.
If somebody had sequenced your genome some years ago, you might not have made the grade in some way.
I mean, that's true. I would hope the society sees the benefit of diversity, not just ancestral diversity, but in our abilities. There's no perfect person.
Despite imperfection, Church has co-authored 527 scientific papers and holds more than 50 patents, proof that great minds do not think alike. The best science can tell, it was about 4 billion years ago that self-replicating molecules set off the spark of biology. Now, humans hold the tools of evolution. But George Church remains in awe of the original mystery, how chemistry became life. Is the most amazing thing about life, then, that it happened at all?
Yeah, it is amazing in our current state of ignorance. We don't even know if it ever happened ever in the rest of the universe. It's awe-inspiring to know that it either happened billions of times or it never happened. Both of those are mind-boggling. It's amazing that you can have such complex structures that make copies of themselves, but it's very hard to do that with machines that we've built. So we're engineers, but we're rather poor engineers compared to the pseudo-engineering that is biological evolution.
Cancer has outwitted scientists and doctors for decades. More than 1,500 people still die of the disease every day in this country. But scientists will tell you they have learned more about cancer in the last 5 years than ever before. And no one is more optimistic about what that will mean for patients than Dr. Patrick Soon-Shiong. He's been called a genius, a showman, an innovator, and a hypester. He's also the richest man in Los Angeles, a doctor and entrepreneur who's worth an estimated $11 billion. Soon-Shiong was a respected surgeon before making his name in the cancer world by developing a multi-billion-dollar drug that few initially thought would work. He now wants to disrupt the conventional way we treat cancer, and Soon-Shiong is overflowing with ideas on how to do it.
That was what I call feed the tumor.
Give Dr. Patrick Soon-Shiong a whiteboard and a few markers, and like a mad scientist, he'll diagram how he thinks cancer can be beaten. He wants to attack on multiple fronts, and is confident there is a pathway to the cure. For 45 minutes, he outlined his vision, from beginning to end.
This is a crazy looking board.
Well, this is what goes in my head. I mean, it's like bursting. It just has to get the stuff out.
Are we looking inside your head?
Yeah, I think so, a little bit.
How long before we get here?
I'm incredibly encouraged to say that we are on the path. And the technology to actually do all of these things is not just hypothetical.
Technology is the main weapon Soon-Shiong is deploying against cancer. In October, at his company's headquarters in Los Angeles, final tests were being run on high-speed tumor genome sequencing machines that Soon-Shiong is convinced will unmask the molecular secrets to cancer.
And for the first time with this technology, we can watch it, catch it, outsmart it, and play chess at this multidimensional level.
To understand the significance of what Soon-Shiong is touting, it's important to know what cancer is.
A cancer is not what people think, cells growing. Actually, a cancer is actually the inability of the cells to die.
The key is figuring out the genetic mutation or glitch that prevents cells from dying a natural death. Soon-Shiong's hope is to provide patients with the precise genetic mutations that fuel their cancer, regardless of where tumors are found in the body.
The mutation that happens in lung cancer could be the exact same mutation that happens in the breast cancer. So you need to treat that patient based on its mutation, not on its physical anatomical location.
That's a big idea. I mean, the idea that the breast cancer specialists are looking for breast cancer mutations, and they may be missing the ball.
Absolutely.
A lung cancer drug could work on breast cancer, for instance, if the mutation is the same. The concept of doing away with labeling the disease by where it's found is not unique to Soon-Shiong, but it is a tectonic shift in the fight against cancer, the notion of classifying a cancer by its mutation.
Imagine reclassifying cancer and having people conceive and understand that cancer is a slew of rare diseases. So I'm very excited because we are going to create this revolution.
And what's it going to mean? I mean,
well, it's going to mean that you have a better shot at having a better outcome and having a quality of life and actually turning cancer hopefully into chronic disease.
That's very optimistic. Realistic as well?
I think so, very much so.
Sun Xiang has appointed himself to lead this revolution. Cancer genome sequencing is not new, but what's different about Sun Xiang's project is the scale. He has spent nearly $1 billion of his own money to build a massive infrastructure run by supercomputers to find every single genetic mutation that could drive cancer. This is Sun Xiang's plan. A patient anywhere in the world has his tumor biopsied. The tumor cell's complete genetic map is then created all the way down to the proteins that are produced. What only recently took months can now be done in a day. Ultimately, personalized information for each cancer patient would show up in the palm of his hand.
This is the baby.
That's it, huh?
It'll be the world's first browser of the cancer genome. So think about that. You'll be able to fly through to get to the single letter that's mutated.
He's teamed with BlackBerry to produce a device that will identify for patients and doctors what they need to make more informed decisions.
At the end of the day, someone has a tumor, they could find out the complete analysis of that tumor and what the perfect drug is to treat it?
Correct. And that's what's so exciting. It's not the end of the day. This is what we think we can bring to the world now.
But some in the cancer world fear Sun Xiang is getting ahead of himself, that he's declaring victory before any of this has been proven to work consistently.
It's show me the money, show me the data, show me that it's true.
Dr. Derek Raghavan, a renowned oncologist and researcher, is president of the Levine Cancer Institute in Charlotte, North Carolina.
Dr. Sun Xiang says, look, if we can figure out which mutations are driving a cancer, we're going to be better able to find the drugs that can treat that cancer. Is that a fair theory? Has that been proven?
Yes, I think that's a fair theory. But to say I can throw a tumor into a gizmo, and that gizmo will tell me the answer in a few minutes, and everything will flow from that, I don't think we're there now. I don't think we'll be there next year. I think there's just too much hard, complex science that has to be done before this is state of the art. But it's a very cool idea for the future.
The vast majority of mutations are actually not a threat. So to figure out which mutations are dangerous, Sun Xiang is going back in time.
This national treasure. Wow.
In the basement of the John Wayne Cancer Institute in Santa Monica, California, decades of cancer tissues were stored by scientists in deep freeze vats. Now Sun Xiang wants to use technology that didn't exist back then to map the genomes of these thousands of tissues in order to look for critical patterns.
So even after a patient died, their samples were stored here. They can go back, say, oh, they had this mutation, and now we can explain that this mutation actually leads to death, and other mutations may not.
That's exactly right. And ask the question, why did this patient live and why did this patient die? Why did this treatment work? Why did that not work?
To make any of this work, Sun Xiang believes you need to upend the way cancer drugs are developed. He started a biotech company to try to dramatically ramp up production.
I know it sounds an audacious goal, but you need to actually develop 20 to 30 drugs a year to actually get ahead of this game.
Right now it takes a few years to create a single drug, and you're talking about 30 drugs in 1 year. Is that really feasible?
I think that's where we have a challenge in the pharmaceutical industry. We actually need to change the way we actually develop drugs now.
Sun Xiang is impatient with the pace of drug approvals. In the early 90s, he invented a drug called Abraxane that treats pancreatic, lung, and breast cancer patients. But more than a decade passed before the FDA approved it.
The problem is, for cancer, however, we don't have that time. You know, if you have pancreatic cancer, you have 2 months if you have metastasis throughout your body. The war against cancer is a war against time.
Sun Xiang is also frustrated with what he calls the trial and error cycle of cancer care.
The truth of the matter, we treat cancer today, we guess. We take what we call the average results, put it on you and see if it works. If it doesn't work, we'll try another drug. If it does work, we'll stop the drug. When you look back 10 years from now, it's almost barbaric.
The 62-year-old native of South Africa can afford to be outspoken because of his immense wealth. He doesn't need to rely on the government or big pharma for funding. Sun Xiang is certain what he terms the dark age of cancer treatment is nearly over, and the Enlightened Age is about to begin.
What will the average person note about the Enlightened Age versus the Dark Age?
The treatment doesn't need to be painful. Metastasis doesn't need to be a death sentence. Cancer could be a chronic disease and treated towards the cure.
While the oncology world may cringe when he boasts, as he's prone to do, patients see him differently.
The established community doesn't like false hope. But if you have a terminal disease, like I do, you want some hope.
David Roy was diagnosed 2 years ago with stage 4 metastatic pancreatic cancer. He was given 4.5 months to live and told to settle his affairs. He called Dr. Soon Shiong, whom he had met on a plane years before. Soon Shiong recommended a UCLA oncologist who devised an unusual therapy that combined Abraxane with other cancer drugs. Then Soon Shiong had Roy's tumor genome sequenced. Based on those results, Roy is now taking part in a clinical trial involving another front in cancer treatment.
That's the T cell, and that's the cancer cell.
It's called immunotherapy. Soon Shiong is not the only one working on it, but he was anxious to show us why oncologists believe it's a promising field. A time-lapse demonstration of how T cells, which our immune systems naturally produce, can attack cancer cells.
That's a cell that's actually gobbling up the cancer cell. This cell will grow in size and the cell will decrease in size. So watch. So here's the T cell gobbling it up. Here's the cancer cell. And
that's amazing. So you're literally watching cancer cells die here.
Correct.
If you find these T cells and you're able to isolate them, is the idea then that you could, they do the job. You can come out and grow them, proliferate them, and put them back into the body?
Exactly.
Even though it's been 2 years now since David Roy's original diagnosis, he's realistic about his chances of survival. But he's convinced Soon Shiong and other scientists are on track to dramatically decrease cancer death rates in the not-too-distant future.
I'm not sure that, it'll happen fast enough for me, but I have every confidence that my children and grandchildren won't be concerned about the things that I'm concerned about. We are on the edge here of going from the oil lamp to electricity, and it is going to happen.
Soon Shiong's most provocative idea, though, centers on how cancers may become metastatic.
He believes chemotherapy works best when administered in frequent, low doses, and that in some cancers, the traditional method of blasting a tumor with heavy doses of chemotherapy may actually be counterproductive, because it could induce cancer cells to escape the hostile environment, enter the bloodstream, and find a new home.
It's on the move. It's on the move. And it's looking for another place to land. So circulating tumor cells in the blood is the next frontier. So here, those are the circulating tumor cells.
That's incredible.
If cancer spreads, the likelihood of survival decreases dramatically. So before individual rogue cancer cells fan out and form new tumors, Soon Shiong wants to detect them with what are known as liquid biopsies. A person's blood sample is put through this biochip that separates normal blood cells from heavier circulating tumor cells. This is a view inside the biochip as the tumor cells are being funneled to the top.
And if we can now monitor the cancer cell in the blood, we then have the path to getting this and winning this war. We never had those paths before.
After pulling out the circulating tumor cells, scientists can take them back to the genome sequencer to look for new mutations that made them resistant to the initial treatment, and hopefully find a new drug to treat it. It's yet another angle Soon Shiong is taking to disrupt cancer.
You get genomics, you have circulating tumor cell, liquid biopsies, death by T cell. Why are you the one taking all of this on? Why, these are lots of different types of things.
Right. Somebody once said to me, Patrick, you're all over the place. And I said, you have to be all over the place because I'm trying to fight this war from all over the place. Because you can't, there's no one single magic bullet.
It's difficult to imagine that 10,000 years ago, right here in North America, there lived giant animals that are now the stuff of legends. Mammoths and mastodons, ground sloths, and saber-toothed cats. They and thousands of other species have vanished from the Earth. And today, partly due to the expansion of one species, ours, animals are going extinct faster than ever before. The very definition of extinct means gone forever. But what if that didn't have to be? As we first reported in early 2010, scientists are making remarkable advances that are bringing us closer than ever before to the possibility of a true animal resurrection. Who wouldn't be dazzled by an animal like this, the woolly mammoth, or the saber-toothed tiger, the Irish elk, the giant sloth? Today they exist just as bones in museums, alive only in our imaginations and the recreations of artists and filmmakers. But what if that could change? In the age of DNA, we now know that these vanished creatures, like all life on Earth, are ultimately nothing more than this: sequences of the 4 letters A, C, T, and G that make up the genetic blueprint or code of life. The codes for extinct animals were thought to have died along with them until recently, when machines like this one at the Smithsonian's DNA lab started working magic.
Just the study of ancient DNA only broke onto the scene 20 years ago or so. The idea that we could harvest DNA from extinct creatures, from fossil bones, learn something about the past.
Sean Carroll, a professor of molecular biology and genetics at the University of Wisconsin, says that like so many things in the field of DNA, the progress has been staggering. One surprising discovery has been the value of ancient hair. Scientists recently discovered that the hair shaft seals DNA inside it like a biological plastic, protecting it and making hair a rich and plentiful source of genetic information. Does that mean that you can take extinct animals? I mean, there's hair in museums,
right? Yeah.
And get the genetic sequencing.
Possibly, and especially if those animals were preserved in any way, there's a good prospect of that. It's sort of like CSI. You know, how good is this forensic material? Can you get good DNA information from older and older and older material? That's pretty promising.
So dusty old specimens that have been tucked away in the drawers of natural history museums like the Smithsonian are suddenly potential treasure troves of genetic information. Just a couple of years ago, using only a few clumps of woolly mammoth hair, scientists at Penn State were able to extract enough DNA fragments to figure out most of its genetic sequence, making the woolly mammoth the first extinct animal to have its genome decoded. Which raises the question of whether resurrecting one of these creatures is really possible. Scientists say one option would be genetic engineering. Take a living animal that's related to the mammoth, like the elephant, figure out all the places where its DNA differs from the mammoth's, and then alter the elephant's DNA to make it match. That's not possible just yet. But there may be another way: cloning. Is it possible that we're going to get the full DNA of the woolly mammoth and be able to clone it?
Yes, I think we'll be able to get much, if not all, of the woolly mammoth DNA. And the great advantage there is that a lot of the specimens are in permafrost, so they've sort of been conveniently frozen for us, which preserves DNA, preserves tissue better.
But for cloning, just knowing the DNA sequence from hair isn't enough. You'd need an intact mammoth cell, which Carroll says will be difficult to find, but not impossible.
It could be a skin cell, it could be any particular cell that hopefully has been preserved well enough, stayed frozen for thousands of years, and to transfer the nucleus of that cell into, for example, an egg of an elephant.
And they're close.
Close enough
that
maybe the elephant could serve as a surrogate mother.
It's called interspecies cloning, implanting DNA from one species into the eggs of another. And anyone who wants to try it with a mammoth or anything else would be well served to pay a visit to Dr. Betsy Dresser in New Orleans. Tucked away on 1,200 acres of land that seem part Serengeti, part high-tech medical facility, she and her staff at the Audubon Nature Institute have been working quietly for years on the science and the art of interspecies cloning. And she'll be the first to tell you that even with living animals, it isn't easy.
You don't just
clones themselves, and then all of a sudden, you have a baby.
Yeah.
I mean, there's so many scientific steps along the way, knowing everything from hormones to the proper surrogate to, length of pregnancy.
Length of pregnancy?
Yeah, because see, we don't know how long a woolly mammoth, the gestation period. We can guess, but we don't know really.
But Betsy Dresser's work on interspecies cloning is focused on the future, not the past. Rather than trying to resurrect extinct creatures, her goal is to keep the animals we have today from going extinct tomorrow.
I feel like we're in the emergency room of the wildlife business, really. I don't want to see elephants in textbooks or, the way we see dinosaurs. We're going to lose a lot of species if we don't do something about it.
Dresser and her team are trying to increase the populations of endangered animals by putting their DNA into the eggs of their non-endangered relatives.
And this cat's going to act as a surrogate mother. And so here's the surgery.
On the day we visited, they were laparoscopically removing eggs from an ordinary house cat, then sending the eggs down the hall to have the house cat DNA literally sucked out of them.
Oh,
tell me what's happening.
What she's doing is she's removing the DNA from this domestic cat egg.
Wow.
And she can see it by what we call fluorescing it. It becomes just very blue. And so now she knows where it is, and now you'll see her go in there and be able to remove it.
She's taking out all the genes.
Right.
Once the house cat DNA is out, that's it being deposited outside of the egg. They will replace it with the DNA of an endangered Arabian sandcat, a completely different species gathered from a tiny piece of skin.
And there you see it being inserted into the domestic cat egg.
And you made that from just skin?
Just from skin cells, right.
An electrical pulse starts the egg dividing, and if all goes as planned, the now sandcat embryo will be put back into the domestic cat to grow to term. It's worked before with African wildcats. These two are both interspecies clones, so normal, they even mated the old-fashioned way and produced kittens.
8 kittens altogether. We had a couple litters.
And they're totally healthy, and they're African wildcats.
Totally African wildcats, totally healthy. And it said to us, hey, this works. And now that we know we can do it, we can say to the world, these animals do develop, they do reproduce naturally, and we can use this as a tool for endangered species.
Is she hissing at us?
Yeah, she's hissing at us.
And Dresser is working her way up. Her next interspecies cloning project will use this non-endangered caracal cat as a surrogate mother for an endangered lynx. And after that, the eland antelope as a surrogate for its endangered cousin, the bongo. You know, there are still people who get nervous at the idea of cloning. They think there's something wrong about it.
And I'll tell you what, if I have to choose cloning or extinction, I'm going to choose cloning. But I want to be darn sure that I know how to do it. And if we don't do it while we have the animals now to be able to learn how to do it, then we're not going to have a choice. It's not going to be an option.
So to keep her options open while she's mastering interspecies cloning, she's also putting as many animals as she can on ice. Literally. Dresser is the keeper of a new kind of zoo, a frozen zoo, where she's collecting tiny skin samples from thousands of different animals representing hundreds of species, and is storing them at 343 degrees below zero in tiny canisters inside these tanks filled with liquid nitrogen.
We've got lions and tigers, we've got gorillas and rhinos, we've got little frogs, all of the animals that people know in zoos.
From this size to this size.
To this size, exactly.
So how long can a piece of skin be viable?
We think these cells can sit here for hundreds, maybe thousands of years.
So if any one of these animals were to go extinct, you could bring them back.
In theory, I believe we can.
In other words, it's kind of a Noah's Ark.
Yeah.
It's not a zoo, it's an ark.
It's an ark, truly.
Do you think we're at the stage where we should be taking every single wild animal, even if they're not endangered, and putting them in a frozen zoo?
Yes, I absolutely do.
Every single one.
What have we got to lose? I think we should put every species in that we can while we have the opportunity.
Which raises the question, with so many living animals today threatened, why think about resurrecting extinct ones like the mammoth? to bring the woolly mammoth back, I mean, we don't have enough space for the big animals we already have.
These projects like the woolly mammoth, they inspire people to think about the meaning of what we're doing here. And why would you invest years and years of your life in trying to bring back a woolly mammoth or taking care of them if you did?
That's an excellent question.
I think it would fire up people's imaginations. And I think there's a 9-year-old girl watching this program and listening to this saying, that's what I want to do. I want to bring back these creatures that are extinct, or I want to protect creatures that are now threatened from going extinct. So in many ways, I think the woolly mammoth can sort of be a poster animal for a general effort of being more conscious of our activities on the planet.
No one has yet found the intact cell it would take to resurrect that poster animal. But in Siberia 4 years ago, a reindeer herder discovered a remarkably well-preserved 1-month-old baby mammoth that had lain frozen in permafrost for 40,000 years. Its DNA was in better shape than any previously found, raising hopes that between new finds and new technology, it may just be a matter of time. Betsy Dresser stepped down as director of the Audubon Research Center recently to work on a book about endangered species and new technology. She continues to consult on the center's work, which is ongoing. You don't expect to hear that some of the most cutting-edge biotechnology is now part of the elite game of polo, the ancient sport of kings. But on a trip to Argentina last December, we went to a big polo match and discovered that several of the champion horses on the field were clones. It's a big day in Buenos Aires, the final match in this year's World Cup/Super Bowl of polo, called the Argentine Open, with the usual pageantry, the tango included. In polo, the horses, called ponies, are just as important as their riders. The two teams are La Dolfina in white and Ellerstina in black. Each team has 4 players who ride as many as a dozen horses during matches. All of the players today have reached the highest ranking in the sport, a 10-goal handicap. The player generating the most interest is the man in the blue and white helmet, Adolfo Cambiaso. He's led his team to victory for the last 5 years. At 42, he's the Tom Brady of polo.
I love the sport that I do. I love polo. I love horses. And so I try to be the best.
You are #1 in the world in your sport. That's stunning, isn't it?
It's strange. When they say it to you, you don't feel like it, but...
How long have you had this title? How many years?
22 years.
You've been the best in the world for 22 years?
That's what they say.
At age 25, Adolfo decided to create his own polo team called La Dolfina and a breeding business from scratch. Today, he has nearly 1,000 horses that are fed a special diet of plants and grasses grown on his massive farms.
If they have a little pain somewhere, I did a swimming pool for them, just to swim.
A swimming pool for the horses, where they do laps and stretch out their sore muscles. And they like it?
They love it.
They do,
They love it. They jump on. It's amazing.
His most prized horse for a long time was named Aiken Cura. But at the Argentine Open 12 years ago, Aiken Cura's leg was broken, and Adolfo was devastated.
More than anything, I say, save this horse.
He was your favorite?
Yes.
But the horse could not be saved. Before they put him down, Adolfo made a fateful decision. He asked his veterinarian to save some of the horse's skin cells. He thought that one day he could bring Aiken Cura back to life through cloning.
I was really sad, and I said, cloning should work.
How did that come into your head?
I don't know. I decided to keep some cells from him, just in case years later, cloning is normal.
he remembered Dolly the sheep, the first cloned mammal. Since then, scientists have cloned cows, pigs, goats, and in 2003, the first horse. Biologist Adrian Muto, one of the first scientists to clone in Argentina, showed us the process. He starts with an egg extracted minutes earlier from a mare.
You can see here, this is an egg, and with that needle, we eliminate all DNA of each egg.
Next, he replaces it with the DNA of the horse they want to clone.
The next step is introduce again into the egg the needle. This is the DNA into the egg.
You did it?
Yeah. This is our cell, and this is the egg.
And that's it?
That's it.
The new embryos are then incubated for 1 week. No sperm has been involved.
We don't need sperm.
There's no male.
Yes, no male here. Only me.
But that's incredible. So it's not a male-female reproduction at all.
Yes.
You're just taking a cell from whichever. It could be a mare or it could be a male
female
horse.
Yes.
And you're putting it in this egg.
Cell into the egg.
No sex at all.
No. Poor horses.
The incubated egg is implanted in a surrogate mare who gives birth to the clone, like this one that's 3 weeks old. Cloning represented a business opportunity to this man, Texas oil heir and polo enthusiast Alan Meeker. He had long dreamt of building a fleet of champion horses, and now had a way to do it.
I did some short math, and I realized it would take 50 years and about $100 million to do what I wanted to do. And I thought, well, why don't I just clone a bunch of horses, really, really good horses?
In 2009, Meeker founded a horse cloning business, and a year later licensed the technology that was used to clone Dolly the sheep.
Okay, now I need to find the best horses. So I put together an idea of licensing the genetics from the very best breeders in polo. I knew some people that knew Adolfo.
Was he considered one of the best breeders? Not just the best players, but also one of the best breeders?
Breeders and owners of horses. His horses were performing better than anyone else.
When Alan first approached you about cloning?
I say yes the first day.
Immediately?
Yes.
And guess what, you said, Alan? Guess what I have?
Yeah, I want to tell him that I have cells from a horse that I really love, that I would love to clone.
It took a while to get it right. One attempt failed, but after 2 years, Adolfo got his wish. The birth of a clone of his beloved Aiken Kura, who grew into this magnificent, healthy horse, almost exactly like the original in strength, athletic ability, and temperament.
When I saw him, I couldn't believe it.
Did you know by just looking, and of course it was a little foal at that point?
Yes, it was. But
did you
to
could
make
tell?
sure,
Yeah.
I took some hair from him, and I bring him back to Argentina to do the DNA.
To double check.
To double check. It was him.
At the same time, he decided to clone another horse, his biggest star, a mare called Quartatera. Now 17 years old, the original has been playing polo since she was 4, a year younger than most polo ponies, simply because she took to the game so quickly.
I think she's born to play. You know, there is those horses in life, or like soccer players, like Messi, it's not many.
Like you.
No, I don't know, no. But when I'm saying this horse is amazing.
He took us to the barn where Quartatera lives with 8 of her clones.
You see those 2 little points?
Yeah.
From these little points is where you make all these horses.
This is where they took the cells
Exactly.
to make the others.
Because of her, you get all these ones.
And that was what you were thinking.
Yes,
I'm
that
gonna
was my dream, but
clone
everybody was
the best. Clone
saying
the
that I
best.
was crazy, and I like it right now because I'm having a good time hearing those people.
Yeah, they're saying he's not so crazy
It's not
anymore.
that crazy, yeah.
And look what he's done. In 7 years, he and his partners have created more than a dozen clones of Quartatera.
It's incredible for me. I never lose my, wow, this is my production. This is my equine daughter.
Dr. Muto, who was hired as the lead scientist in Adolfo's cloning business, took us to see the Quarta Terra clones he thinks of as his children.
This is Quarta Terra number 5.
Oh my God.
This is number 4, number 3, number 9.
Oh my God.
Number 6.
You can tell which one.
Yeah,
You're not reading
because
anything.
I know her, but they are all clowns. Right now we have 14.
Just from Quarta Terra.
14, and next year 10 more, and 2019 10 more.
In all, they have produced more than 100 clones from several of their best horses. In each case, he said the clones are strikingly similar to the originals in disposition, athletic ability, and appearance, but not exactly. For example, the Quarta Terra clones all have white markings, but with different shapes and in different places, some on the face, some on the ankle. But all the Quarta Terras seem to have inherited the original's calm, self-contained personality. So the genetics include this temperament.
Yes.
And do the clones get along with each other?
Yeah, because they live together all year alone. So from here they go to the farm together, then they move in blocks. If you take one out of them, they're looking for it.
They missed the one that you take out.
Yeah.
Do the clones have any special health issues?
No.
We talked to scientists at the National Institute of Health and were told there was no evidence that cloned animals suffer disproportionate health problems, though they have a slightly higher infant mortality rate. At first, many of Adolfo's cloned embryos died during gestation, but they refined their technique and now tell us they have an 85% successful birth rate and have not experienced any health problems. So as far as you are thinking, they are exactly the same in health, longevity, ability to play the game, all of it.
Similar, not exactly the same.
What are the differences?
There is some that are a little bigger, some eat more, some eat less, or they move a little bit different, but the mind are really similar. The good thing about it, there are machines, all of them.
Machines, that's polo talk for horses that never quit. But how would they perform in competition? At the final at the Argentine Open, Adolfo gambled that his Quarta Terra clones would be as good as the original. And for the first time, he rode them almost exclusively. That part of the story when we come back. Regulators of thoroughbred horse racing worldwide have taken a firm stand against cloning. But there is no such prohibition in polo, and so cloning is spreading to teams beyond Adolfo Cambiaso's. It raises some thorny questions. Does cloning give a team an unfair advantage? Is it ethical? And where will it lead? In December, at the final match of the Argentine Open in Buenos Aires, one team rode clones while the other refused to. The competition was as much about the merits of cloning as it was a sporting contest. Out of more than 850 professional polo players, Argentine native Adolfo Cambiaso, wearing the blue and white helmet and the jersey marked #1, is the best player in the world. He's held that ranking for 22 years and is now leading a cloning revolution. He's cloning his best horses. The one he's riding is a clone. He's competing on them and winning. When you're on one of the clones playing, is there a special feeling knowing that, this is something that was your idea, you brought it to life?
At this stage of my career, the last couple of years for me to play and prove that the clone works and play with Quarta Terras and everything is an extra motivation for myself, for sure.
I don't know that you need extra motivation.
Yeah, I do.
He's created 14 clones of Quarta Terra, his very best horse, a 17-year-old mare who is fast, easy to direct, and can turn on a dime. She was honored last year as the best polo horse in history. Her clones seem to be just as gifted. Are they all as good? I want to call her Mama. I don't think that's probably the right word.
The original.
The original.
Yeah.
Are they all as good?
I already won 2 Argentine Opens. with the clone. So they will end up being as good as her, I think.
Quattatera's clones are identified by numbers.
Shouldn't they have names?
They have names.
Well, they don't.
Quartetera.
Yeah, but...
Quartetera 1, 2, 3, 4.
Was that your idea?
Yes, because I believe that she is a quartetera. All of the ones that I write, they're quarteteras.
So you actually think...
She's quartetera. When I play,
so 6 is quattetera,
it's quartetera.
And when you're on 9, it's the same thing?
Quartetera.
But she's an individual.
Yes, but the DNA is a quartetera.
Yeah, but when you have identical twins, they each get a name.
But this is not twins, it's a clone.
They can now create 100 clones a year, and they're using them in Adolfo's already successful breeding business. They mate the clones with champion horses and sell their foals for up to $250,000, but they never sell the clones.
You sell the clone, you sell the blood, you sell the line, you sell the DNA, you sell everything if you sell the clone.
We keep the key of this genetics. And this was I think the good business to make that decision in the past.
The idea of never selling the clones came from Ernesto Gutierrez, a shrewd Argentine businessman who became a third partner in the cloning venture. The cloning operation was set up here on his 500-acre property outside Buenos Aires that includes 3 polo fields and a nursery where the clones are born. They are carried by surrogate mares who treat them like their own.
Are these all cloned babies in
All
here?
clone babies, yeah.
And these are the surrogate mothers. And does the mother think it's totally her baby?
Totally. Look at that.
Gutierrez took us back to see the newest one, that 3-week-old clone of Quattatera, who has her own nurse.
Sweet. Oh, and frisky. Oh, look at that. Oh, my. Oh.
Not everyone in polo thinks cloning is a good idea, including Adolfo Cambiaso's main rival and opponent at the final of the Argentine Open.
There's a lot of guys cloning, but I think that they have to be careful, because the thing is that they're opening too much, I mean, the
Pandora's
business.
box. They've opened the lid and all the problems
Exactly.
come out.
Facundo Pierres is number 2 in the world, right behind Adolfo. He showed us what he can do, like dribble a 3-inch ball in the air while galloping down the field 20 miles an hour. He's the captain of Ellerstina, an old-school team made up of 3 brothers and a cousin. They're committed to keeping it a family enterprise.
Do you ever get angry at each other and...
Yes. Yes, but in a good way. Never bad.
His team is headquartered at another sprawling estate where they operate a multi-million-dollar breeding business selling foals and embryos. They believe they can produce better horses through their breeding practices by mixing the DNA of 2 different horses rather than by replicating just one.
We want to keep it this way, and what we have here is amazing.
In polo, what's more important, the horse or the player? I was told that it's 80% the horse.
Yes,
Sorry.
I agree.
No offense.
No, I agree. I totally agree. I think that the horse is, but of course you need to have a little bit of talent and ability and experience in the head,
Facundo's team, Ellerstina, has made it to the finals the last 4 years, but lost each time to Adolfo Cambiaso's team. Fueling the rivalry on the field is a bitter history between them. Adolfo played on the Ellerstina team for 9 years.
Because of what happened, that I left Ellerstina, the rivalry is
Intense.
there. Yeah.
To this minute.
Yeah.
Do you feel it?
But it's fun.
Oh,
you
you
gotta
like
have
it?
rivalry to be a better player too.
There's more. Before he left Ellerstina, Adolfo bought Quattatera as an embryo from the Pierres family, the very horse he is now cloning to compete against them.
I was lucky to end up with Quattatera,
You cloned from the best horse in the world.
but she born in my farm. I create her.
There are people who object to cloning on religious grounds or on moral grounds. So what is the answer when people challenge you? When they say, man should not be doing this because of these difficult spiritual questions?
I don't see it wrong, to be honest. I'm just doing something to improve my game, my sport. And I think the cuarteteras, it did improve my game, my sport, and I'm not going farther than that.
But is there an unfair advantage in terms of the game, in terms of the sport?
No, because everybody is able to clone. Now everybody is kind of trying to start cloning. So the advantage is that I did it 7 years ago.
So in 30 years, people will still be riding cuartetera?
Yes.
So it could go on forever?
Yes.
Alan Meeker, the Texas businessman who's Adolfo's cloning partner, is well aware of the controversy around cloning technology in the U.S. and the ban against it in thoroughbred horse racing.
Is a really good polo player, does he have an unfair advantage if he's on a clone of one of the best polo horses ever?
Of course. Horses are 80% of the game anyway. So if Facundo Pierres finds a horse that is better than cuartetera, then he has an advantage over his competitor.
But he'll have only one, and that horse will get tired, and they'll have to switch to another horse in the game, whereas Adolfo will have 8.
Right.
So it's still an advantage.
Right.
Is that fair?
Under the rules, it's fair. There's no,
I know, but
there's no restriction on...
But sportsmanship, but just the nature of the game. Has this changed the very essence of the game of polo?
No, I think what it's done is it's probably raised the bar.
You're going to have to clone.
Could be, yes.
Do you have any moral problems with cloning a human being?
Yes. I disagree with it. I know a good reason, lots of good reasons, to clone body parts like hearts and lungs and pancreases. If it could be done in a productive manner, that can save lives. But I've been asked by some of the wealthiest people on planet Earth to clone a human being. And
you have?
Absolutely. And the answer is always a resounding no.
Well, they must have a reason.
And they won't give it to me.
They don't tell you why.
No.
I'm thinking if science can do it, science will do it, and maybe one day, there'll be clones and we'll laugh at all the people who are questioning the morality of it now.
Someday someone will do it.
Yes.
And we will either laugh or we will cry. But I'm not going to be the one to take that leap.
It could be done today.
Yes.
I assume there'd be a big difference between a horse and a human. Lots of differences.
Surprisingly little. Yeah, surprisingly little.
At the final match at the Argentine Open, Adolfo's team and the clones were expected to win. But 7 minutes in, Facundo's team was ahead, 3-1. Adolfo's team fought back. At halftime, the score was the cloners 7, the breeders 6. It was so tense that at times it was as quiet as a tennis match. The end of the game was thrilling. As expected, Adolfo's team was ahead 13-10. But then Facundo's team, in a final blast, came back to tie the match.
I never think I'm going to lose. I never.
Well, we saw you right before the overtime.
Yeah.
And here you are, like that.
In that moment, I was trying to think which is the best horse for that moment.
He debated, should it be Cuartetera 9 or 5? Finally, he picked number 6. In the first minute of the sudden death overtime, Facundo's team lost control of the ball. Adolfo's team recovered, and Adolfo, on his mighty Cuartetera 6, outran everyone and whacked the ball, setting up the winning shot.
Vanero, y llega, y el gol de la Dolfina para ganarlo en suplemento.
Watching, you had to wonder, was it the clones or the world's best player that made the difference?